Recording now. Chapter 10. Let me know if it cuts off again in 2 seconds. Chapter 10, all about DNA. So there's some famous experiments that would make good essay fodder, just to warn you when we get to those. What does it look like? What does it do, basically? We're going to add a tiny bit, which I think will help explain some of the material that's not in the chapter that you need to know about. Very little, so let's get into it. So they knew what DNA was a long time before they knew what it did. So by the 1800s, they knew of DNA, they could isolate it, and then even early 1800s, they knew what it was made of, nucleotides, these different A, G, C molecules. Then let's kind of switch gears and talk about inheritance. They didn't know DNA did inheritance until like the middle of the 20th century. But Mendel in the 19th century, he'd been breeding pea plants, and he discovered that you could predict what the children would look like with deadly accuracy. 75% chance of them having flowers that are purple instead of white, and the grandchildren, and dominant, recessive, and these alleles and traits. And so they thought they had inheritance all squared away and neatly tied up in a neat little bow. They knew why children look like their parents. They were giving them some sort of information that made them grow that way. They knew how to predict it. But we're going to talk about some moments in this chapter and important things other than that. So by the early 1900s, we had this chromosomal theory, right, that said chromosomes are the genetic material of inheritance. But it was just a theory. Again, nobody really... They knew what DNA was, and it was on chromosomes and stuff, but they didn't really think it controlled your inheritance. And I'll explain why in a moment. Well, we'll talk about that now. DNA only has 4 variables, A, G, C, or T, adenine, guanine, cytosine, thymine. There's not much you can do with an alphabet of 4 letters. Protein, on the other hand, there's 20 different amino acids and an unlimited number of protein shapes with different functions. So everybody thought to make something complicated, you had to be complicated, right? Darwin's kind of theory of natural selection and simple, elegant concepts making complexity, that wasn't really well subscribed to until the middle of the 19th century. And Alan Turing, or World War II, 1940s, he was, breaking the unbreakable Enigma code, and he was really the father of modern-day computing. He figured out that just using ones and zeros, that was enough to create all this complexity, right? Just an on or an off, a yes or a no. You didn't even need a quaternary, of A, G, C, T. You only needed a binary system to do so. So slowly we started to kind of change our thinking, and we'll look at one of the crucial experiments that help show that the inheritance molecule is DNA, not protein. It's the Hershey-Chase experiment. We'll get to that in a moment. But a little bit more about history, stuff like that. Beadle and Tatum, super important. They're the ones So there's several really key ways to investigate things in life. One way is to break something and then compare it to a non-broken version to find out what it does. Like imagine if you just came to a car. You had no idea what the thing did. You could just like smash one of the headlights, and then compare it to another car, and you're like, oh, that makes the light come out. Or go to the gear shift and just break it off, and notice you can't change. Oh, that controls this. So that's one of the key elements in the way they discovered this, that a gene codes for a protein is by breaking genes and seeing that they didn't make protein, basically. So that's a key way of investigating the unknown. Break it and see the consequences of it by comparing it to a non-broken one. Griffith's experiment. This might make a good essay question. So you really need to understand this. He worked with 2 different strains of bacteria. A deadly one, the smooth version, the S, and a non-deadly one, the rough or R strain. If you injected the harmless strain, the mouse lived. If you injected the deadly strain that had this protective coating and made them look kind of slimy to the naked eye, the colonies growing on a plate, if you injected the deadly strain, the mouse died. What is going on? Why isn't it showing the mouse? What? Okay, I guess this is just the living. Oh, that's the. Okay, so they're doing it in a different order I'm used to. Living, harmless, mouse lives. If you kill the harmful strain, it's dead, it can't hurt the mouse lives. But if you inject the living harmful strain, the mouse dies. That should be yawns at this point. Here's the kicker though. Where these two by themselves led to mouse living, if you mix them together, the living harmless and the dead harmful, if you mix them in the same syringe, the mouse dies. And you're able to culture from the dead mouse, you're able to culture the living harmful. So it wasn't that the harmful bacteria was coming back to life like a zombie. The conclusion from this is that living bacteria could pick up not just characteristics from dead organisms, but inheritable characteristics that they could pass down to their progeny. So this flipped Mendelian genetics upside down. We knew parents could give genes through sex to the children and chromosomes and dominant, recessive. But here was a dead thing influencing the inheritance of a living thing. No sex, no babies, no grandchildren. So this was the first example of horizontal gene transfer. So they didn't know it in the 1920s when Griffith did this experiment. But what happened was, the reason the harmful bacteria could produce the protective shield, it had an extra piece of DNA. It had an extra gene that coded for the ability to make the protective coating. So it's ready to split. Even though these cells were dead, the gene was still intact. Well, no, even though these cells were dead, the DNA was still intact. So luckily, these cells were quote-unquote competent, meaning they could just pick up the DNA. He called this phenomenon transformation. The harmless strain had been transformed into the deadly strain through the acquisition of the harmful bacteria's naked DNA just floating around. Again, the harmful bacteria was dead, but the genes that made it harmful were still intact. And the harmless bacteria just picked up the DNA laterally, not vertically, like when one cell divides and passes on its DNA. That's vertical transmission of genetic material. This is horizontal or lateral transmission. Again, he called this transformation. So they didn't know why in the 20s, but they later figured out how transformation occurred, lateral gene transfer. So that's a key experiment. You know, basically they work with two bacteria, one killed, one didn't. When you mix the two that didn't kill, it did kill, and the harmless bacteria picked up inheritable characteristics from the dead bacteria. I kind of, I might have to read into it a little more. I don't know how well I explain that, but it's an important experiment. Another super crucial experiment, which I alluded to earlier, the Hershey-Chase experiments. So the take-home message from Hershey-Chase, it proved once and for all that the blueprints for making organisms was encrypted in DNA, not encrypted in protein. That all the top scientists in the 1920s, or when did this take place? Somewhere around there. Early 20th century, pre-World War II certainly. All the top scientists thought that whatever parents were giving their children that made them look similar was protein. But this experiment proved once and for all, it's not protein, it's DNA, which again is a simple molecule, but you don't need a complicated system. You don't need this like complicated creator to create things. You can have a bottom-up, bubbling up of complexity from kind of grassroots. So anyways, here's how the Hershey, it's actually two different experiments. Dr. Hershey and Dr. Chase, or I think Chase might have been his graduate student. I don't know. Maybe they were, I don't remember the details, but anyway, two scientists. So basically they grew phages. A bacteriophage is a virus that infects bacteria and only bacteria. So they grew one set of viruses and they added radioactive phosphorus. Remember we said in the last chapter, the video cut off, but we looked at the geological cycles. Phosphorus is key in making DNA, but there are no phosphorus in amino acids, in protein. So by adding radioactive phosphorus to a batch of replicating viruses, they incorporated it into their DNA. They had radioactive DNA, but not radioactive protein. I think that's, yeah. So the radioactive labeled DNA. I would have made that red in this photo to make it easier. And then in a different batch of viruses, they added radioactive sulfur. Again, sulfur is not found in DNA, but it is found in certain amino acids. So this made viruses with non-radioactive DNA, but radioactive protein. A virus, this is a great model. A typical virus is just a nucleic acid like DNA or RNA and a protein protective delivery mechanism, a capsid. So you could either label the protein, or you could label, so in one experiment, they radio-labeled the DNA and left the protein non-radioactive. In another experiment, they radio-labeled the protein and left the DNA non-radioactive. Right? Two separate experiments. When these viruses infected bacteria, nothing radioactive went in the cell. So this proved that the radioactive protein didn't go in the cell. Yet these were able to make virus babies. So the cell received the information on how to make viruses, but it couldn't be made out of protein because the radioactive protein didn't even go in the cell. But in these cells where the DNA was radio-labeled, they were able to make radioactive virus, well, some of the virus babies were radioactive, but radioactivity was found inside the host cell. So this proved that the DNA is entering the cell, but the protein is not entering the cell. So the information must be written in DNA. That's the gist of